and hi. Uh, we are from K2G9. Today we're going to present about active transport. So first I'm going to let Nurul explain about her part.
Hello and hi. My name is Nurul Shafika binti Muhammad Sani and my matric number is 216721. Today we want to discuss about active transport. What is active transport? Active transport is the movement of ions or molecule across the cell membrane from region of the lower concentration gradient to a region of higher concentration gradient. Require a cellular energy to achieve the movement. So hence, this process will require the expenditure of energy, the assistance of membrane protein such as carrier protein. So we look at the structure there. Which is a phospholipid bilayer. Then in the structure we have carrier protein. So the carrier protein is a protein that transports specific substance through intracellular compartment into the extracellular fluid. Carrier protein also the type of cell membrane protein that involve in facilitated diffusion and active transport. So, what is the function of active transport? Function of active transport is used by cell to accumulate needed molecules such as glucose and amino acid. Next, transport of molecules against the concentration gradient. For example, we have here is pumping of sodium and potassium ion across the nerve cell membrane during the transmission of impulse. As we can see in the structure, we have up on the up there we have lower concentration of molecular ion, and then down there we have high concentration of molecules ion. So the molecular ion will be carried through the lower concentration gradient to higher concentration gradient. So that's all for me.
Hello, my name is Hana Maisara binti Nirwan. Matric number 215243. So I'm going to present about calcium pump. So calcium pump is a membrane-bound ATPase which maintain low concentration of calcium ion in cytosol compared to higher extracellular concentration of calcium ion, which at the membrane-bound ATPase in sarcoplasmic reticulum of muscle cell. So action potential which depolarize muscle cell membrane trigger the sarcoplasmic reticulum to release calcium ion into the sarcoplasm. It also will stimulate muscle contraction. So the function of calcium ion in muscle contraction is the calcium ion will bind to troponin, which help to expose myosin binding site on the actin, which allow myosin to bind to actin. The other function of calcium ion in muscle contraction is calcium ion stimulate ATPase activity to hydrolyze ATP to ADP and phosphate group and change the myosin head from low to high energy configuration. That's all for me. Thank you. So for the next person.
Hello, everyone. This is Sabrina. My matric number is 213745, and I will now move on to the proton pump. So this is a type of integral membrane bound enzyme ion transporter. That is, it encompasses the entire membrane, and it can work in both directions for transporting hydrogen ions, that is proton, to the cell and also from the cell, both inside and outside. So when it transports hydrogen ions from inside the membrane to the outside, it requires energy, active transport, and it can also work in the opposite direction, whereby it releases energy. During cellular respiration, in the process of the electron transport chain, it releases the energy. That energy helps the pump to push the hydrogen ions from the matrix of the mitochondria to the intermembrane space. And Nurul, could you please slide down a bit? So another thing about the proton pump is it's essentially found in mitochondrial membrane. So that's why we're talking about the intermembrane protein. Hydrogen ions produced by oxidative phosphorylation in the mitochondrial matrix, using energy from the electron transport, it transfers it outside. Since the hydrogen ion concentration in the intermembrane space is more, so this creates an electrochemical gradient, and this kind of acts like a battery. So when the hydrogen ions wants to move back into the cell, this gradient generates energy. So the proton pump essentially acts like an ATP synthase, where it combines ADP and a phosphate to form ATP. So that's about it. Thank you.
So me, I'm Tristan Regaud, and my matric number is ES03845. So now we will approach the topic of the sodium ion and potassium ion pump. So this transmembrane protein has the ability to transfer the two ions against their gradients. This means that at the beginning, the sodium ions are inside the cells, whereas the potassium ions are... No, it's... Oh putain, c'est l'inverse. So, sorry, I did a mistake. So I repeat, at the beginning, the sodium ions are outside the cells, whereas the potassium ions are inside. And during the transfer of the three ions, the ATP will fix one subunit of the molecules, and the pump will open and transfer the ions. So during the transfer, 3 ions of sodium will go outside the cells, and 2 ions of potassium will go inside the cells. As a result, it will be more positive electric load inside and more negative outside. All of this is possible thanks to the 3 principal subunits of the pump. The alpha one allows the transfer, the beta one hydrolyzes the ATP to produce energy, and the gamma one allows to regulate the reaction by modifying the affinity of the pump with ATP.
Hi, my name is Baudouin, and my matric is ES03808. And I'm going to be more precise about how the pump works in order to create these gradients of charge. So first, the pump is going to bind with 3 sodium ions and 1 ATP that is going to be broken down into ADP plus 1 inorganic phosphate. And the inorganic phosphate is going to remain bonded to the pump. Then the pump will change its shape, its conformation, in order to release outside the cell the 3 sodium ions. And the inorganic phosphates will still remain bonded, while 2 new ions, 2 potassium ions, are going to be linked to the pump and will change again its conformation in order to release the 2 new ions inside the cell. And then the cycle repeats again in order to create this new gradient of charge along the cell membrane. And this whole process allows to create a charge difference that is used for the conduction of nerve influx or nerve impulses. And the main step that involves the pump in those nerve impulses is named action potential. And the other steps are the resting membrane potential, the depolarization, the threshold, propagation, repolarization, and the refractory period. And this action potential can be defined as the travel along the side, along the cell membrane, of this influx, this electrical influx, thanks to the sodium-potassium pump. Thank you.
My name is Anisha Yasmin. My metric number is 214174. I will explain about ABC transporter superfamily. ABC transporter superfamily is a type of carrier protein. It is specific to a single substrate or family of substrates, ions, and sugar. They consist of 4 domains, which is 2 transmembrane domains and 2 cytosolic ATP binding domains. Normal functions of ABC transporter superfamily is first, substrate bind to high affinity sites. Second, allows for 2 ADP to be exchanged for 2 ATP at ATPase site in NBD, which is nucleotide binding domain. 2 nucleotide binding domain dimerize when bound to ATP. Dimerizations allow substrates in cavity to be released to other sites. And lastly, hydrolysis of ATP restores transporter back to initial. So I will explain about large molecule that require ATP. The first one is exocytosis, which is push molecules out of cell. Second one is endocytosis, which cells taking in substances from outside the cell by engulfing them in a vesicle. There are 2 types of endocytosis, which is phagocytosis and pinocytosis. Okay, phagocytosis is called cellular eating. Why? Because cell use its plasma membrane to engulf a large particle, giving rise to an internal compartment called fibrosome. While pinocytosis called cellular drinking because cell takes in the fluids along with dissolve a small molecule. Okay, that's all. Thank you.